Good morning, everybody. I'll get today's lecture started. Today, we're going to discuss somatic cell nuclear transfer. Previously, we have learned that to generate pluripotent stem cells, generally speaking, we have three roads that we can follow. The first is the natural way. You make animals to mate, and then you isolate blastocyst. You can establish the embryonic stem cell, which is the gold standard of pluripotency. And you can do that. That's the natural way. And then we have two other ways that's artificial. Scientists discovered that you can generate pluripotent stem cells by two other ways. The first is called somatic cell nuclear transfer, which is going to be today's focus. And that is going to be the discussion topic of this week's student lab discussion. Group 2 is going to deliver that. I have posted the material. The rest of the class can go to class materials and study the paper that I posted for the discussion as well. And since the scientist who originally discovered this somatic cell nuclear transfer technology was John Gurdon, I hope you will read the story of John Gurdon, which you can find in our textbook as well as search the internet. This is a famous scientist. There are a lot of stories about him. So I want to check if anybody from group 2 is here. If you're here, can you please speak up to let me know that you're here?
We are here.
Okay, that's good. That's good. So I just want to make sure you are aware that I posted the material, and I also expect you to tell us something about John Gurdon, not just the paper, but the life of John Gurdon. This is a legendary scientist who earned a knight for not only the Nobel Prize, but knight from Queen of England to have a title, a Nobel title. And his work is really extraordinary. So make sure that you include something about that, not only about science. Okay. In somatic cell nuclear transfer, what you do is, you take your task here is to reprogram a somatic cell toward a pluripotent stem cell, right? That's your task there. So to do that, you remove the nucleus from an oocyte. The oocyte will have a nucleus, right? You remove the nucleus from oocyte, leaving back just the cytoplasm of oocyte, the egg. And then you take the nucleus of any somatic cell, your liver cell, heart cell, fibroblast, whatever of your choice. And then you just take that cell nucleus. You put that genetic material into an oocyte. In that oocyte, what you have is just cytoplasm. You don't have nucleus. And then the cytoplasm, the proteins, the RNAs, whatever is in the cytoplasm is going to do the magic. It's going to turn the genetic material of a somatic cell to become, the genetic material of a pluripotent stem cell. And eventually, that can lead to development, normal development. You can generate blastocyst and then isolate pluripotent stem cell, or you can allow that blastocyst to develop eventually to give an individual. Okay, at this point, I hope everybody have learned that pluripotent stem cell is almost equivalent to life, to a living individual, because you can always allow that blastocyst to grow, to reach the stage of infant, adult, if you choose to, okay? But apparently you cannot do that in humans. But in animals, in other species, you have that choice. So that's the second way to generate pluripotent stem cell. It's by somatic cell nuclear transfer. And the third way is by just putting some regulatory factors, not necessarily transcription factors, but at the beginning, Shen and Yamanaka used the transcription factors. So you put a few transcription factors into a somatic cell, for example, like a fibroblast, and eventually, after some time, a long process, those fibroblasts will give rise to some pluripotent stem cell. And again, that pluripotent stem cell, can be just the cells, and then it can be an individual if you choose to do so. Okay, so we have 3 ways to generate pluripotent stem cell, and in today's lecture, I'm going to introduce somatic cell nuclear transfer. You heard it many times, but I'm going to include some more technical detail about this. So this is called somatic cell nuclear transfer, shown on the very the left, top left is Sir John Gurdon. And I had the fortune to meet with him a few years ago when he delivered a talk in Hotel Galvez in a stem cell meeting. We used to have a very good meeting for 3 or 4 consecutive years, but later on, maybe because of the attendance was not that great, and then they canceled that meeting. Otherwise, it is one of the best stem cell meeting. And I really hope that, if the meeting's here, I can take you guys to the meeting to listen to some of the best scientists in this field, but we can't do that now. So John Gurdon did this when he was undergraduate. He found that, he studied, this is something he did all by himself. His advisor probably appointed some other topic for his PhD thesis, but he chose to do something that he's interested in. That is, this line of embryo manipulation. So what he used was, I think it was a species called clawed toad. The toad is like a frog. And then he didn't have maybe not the equipment or maybe not the technique to remove the egg, to remove the nucleus from the egg. So what he did is using radiation to destroy the nucleus of the egg. So the egg is still there, but inactivated. The egg is non-functional any longer, destroyed by radiation. And then he took the intestinal epithelium from tadpole, which is at the early developmental stage of toad. It's a tadpole. It took the intestinal epithelium and then he put the nucleus into the enucleated egg. So now the egg gets transplanted with a new nucleus, but that nucleus is from a young toad intestinal epithelium. And he found that egg can develop. It can develop to the 2-cell, 4-cell, all the developmental stages, and eventually can develop into a tadpole, which was really, really amazing. It was in 1960 when he first described this. His paper, I believe, was published in 1961 or 1962. I couldn't remember precisely, but it was around that time. But his work didn't publish in some of the best journals, like Science, Nature, Cell, those are the most, high-level prestigious journals that, as you probably know. But it was in a relatively a smaller journal. The student will present that on Friday's, student-led discussion. Why it wasn't accepted by the scientific community at that time, and as a matter of fact, it wasn't accepted for quite some time. So I would like you guys to tell me why this wasn't accepted, or what could be the concerns. I can give you a hint. It's all things about ethical reasons. Can somebody please speak up? Just speak up. We have a small class.
I thought it would be ethics as well because I mean, like they were afraid that possibly you could make a clone or like repeat like a human being using this kind of cells, even though like they don't, like they might have some issues with that in some ethics, but exactly what I'm not sure.
Okay, so you can
What was the question
answer.
again, Professor?
Now my question here is, this piece of work is great in today's standard. We know this is great, but at the time, and even many years after he first described this work, it's not very well accepted by the scientific community. And as I said, there were quite a few ethical concerns. They want you to discuss this. What do you think could be those concerns?
Could it be a religion maybe? I mean, I know if he's in England or the UK,
Yeah, that's good.
Yeah.
Good. It's a good point. Yeah. Anything further?
I really can't think of anything else other than maybe religion or ethics.
Yeah, I think the first student was Louis, right? Was it
Do
you?
it, yeah.
Yeah, he said that. So just continue. Anybody who want to say anything, because I said it is more about ethical things. If not, I'm going to say something about this. So first of all, this is called. I have 2 points here. One, this is asexual reproduction, because now consider it in a natural way. It's always a male and a female. Not in all the species, but in things that we are familiar with, like mammals, it's usually a male and a female. By mating, the egg get fertilized and eventually give rise to an individual. So in that process, what you have is a mixture of genetic materials from two parties, right? It's always a mixture of two parties. Sperm carries one end, oocyte one end, and then you have two ends. That's the natural way. But here, the genetic material is from one party. You see, the oocyte only give the cytoplasm. You don't have the nucleus from the oocyte any longer. So that cell, the donor cell, can be from a male or female. It really doesn't matter, right? It could be from a female animal, and you just put into the genetic material into an oocyte. Now this is asexual reproduction. First of all, this is not sexual, not natural, and secondly, this is cloning. And previously, when you do a two-party reproduction, basically the nature is telling us to mix the advantage and disadvantage of two parties. Most times it's because of the advantage. It's natural and it does something good. And now this is cloning. Actually, you're trying to replicate something, right? You're trying, like this guy. I don't use human as my example. Like you have two dogs, right? And these two dogs, no matter they're the same. I don't know what's the name for it. Maybe there's, but sometimes you can make it two different dogs, right? And to create a different breed, and not a mixed breed. But here, by cloning, basically, you're not cloning just a breed, but maybe just that individual. You're replicating the genetic material exactly as the donor. So this is called a cloning. You're replicating something. It's not in the natural way. Reproduction is not doing that. It's always a mixture of two parts. So that's the two major things. It is asexual, and also you're trying to replicate. And also people, that's what you guys have alluded to. There is a possibility of reproduction, that you're going to generate some human being, in a very, very weird way. Perhaps not a very good way. You will learn. Maybe that's not a very good way, but others will argue that we don't do that. We're just clone this perhaps animal or even human being, but we don't clone this for a reproduction purpose. We we do this for a therapeutic purpose. So people start give this two names. Like one's reproductive cloning, the other is therapeutic cloning, but the roots are quite similar. The roots diverge at one point, at the point of blastocyst. If you isolated the blastocyst, you won't allow the embryo to go further, and now you can build embryonic stem cells, and then you can use the cells for regeneration of tissues or for other studies, right? But there is that possibility that somebody can create a clone of somebody. There is certainly that possibility there. So that's in my opinion, probably that's why it took that many years for the field to actually recognize Sir Gurdon's work. He had the discovery in 1960, but it was not until maybe 2012. That's about 52 years or so for the scientific community to give the credit that it deserves. He won Nobel Prize in 2012. But I want you to appreciate that there are these concerns. But I guess there are less concerns if you use this technology in animals. So Wilmot, that's the German scientist, Wilmot first used this technology to develop Dolly. You remember in the very first class, I mentioned that. And then Dolly was named after Dolly Parton. And after that, as shown on this table, there were many cloning going on. The table is it's not updated. It's up to 2007, just 10 years of time. You see mouse, pig, cat, cow, goat, dog, all those things get cloned. So why they want to do the cloning? For most part in the agriculture, or I don't know how you call that that business. It's either farming business or pet, companion animals, or for whatever reason, other reasons, they're trying to replicate that. Well, sometimes it's because they just want the same dog who's passed away to come back. Some companies offer that service. You can get your dog cloned. That's a possibility. Looks quite similar, I guess. Much more similar than if you have a natural breeding. So because this is cloning, the genetic material is just from that animal. And sometimes I would say maybe most times people do cloning in animals because they want to select some trait that's really giving quite a lot of advantage, like maybe we have rodeo. I don't know whether this year we're going to have rodeo in Houston or not, but some of the bull, I believe the owner really wanted to get those cloned because they're powerful. They provide a lot of entertainment, so they want that cloned. They don't want to have the bull to mate with a female to lose that trait, because there is a possibility. Okay, if they do cloning, they can keep that. For example, if some cow really produce a lot of milk, you probably want to get it cloned. So in farming and there are needs, basically, what I'm trying to say. So a lot of animals actually get cloned. And you probably read some news or watching television. A lot of people are trying to bring back the mammoth. It's such a wonderful animal, good looking. And I show that picture. The picture is from Royal BC Museum, a museum in Victoria Island off Vancouver. That museum is really cool. If you ever have a chance to visit that island, please do to go to pay a visit to the Royal BC Museum. So what they did there is they have all the artifact of animals shown in the foreground. You know, that's the artifact of animals showing in foreground, but they put something like earth or tree or something around the animal, still in the foreground, and then they paint the background. They did such a nice work. When you step into the hall, you feel like, wow, you come to a forest or open land. You see the animal there. It looks so real. But actually, they just have something on the foreground, but otherwise, everything else is just painting and light. They used it so gracefully. It's really one of a kind. If you ever have a chance, please do go to pay a visit. So, you know, it's ongoing. People are trying to use Asian elephant or maybe even the mammoth residue. They dig it out of ice in Russia, and then they try to extract the DNA and see if they can clone that. They put the mammoth DNA into you know, nowadays elephant and see if the mammoth can be cloned. So you know, this is cloning. It's not the natural way. But then remember that the work was done like in 1962, and a lot of cloning, you know, what's going on in the early days, you know, the first 10 years of 20. And then people are also racing for generating human embryonic stem cells by using somatic cell nuclear transfer. Because as I said, the scientists have already recognized that embryonic stem cells are very useful because, you know, the three scientists and also Gil Martin described that you can extract mouse embryonic stem cells in 1981. And then there was a race that scientists are trying to develop human embryonic stem cells. If you still recall, in the previous lecture, I said that human embryonic stem cells arrived only in about 2007. So that's 27 years after the isolation of mouse embryonic stem cells. But somatic cell nuclear transfer was established in 1960, even earlier than the extraction of embryonic stem cells. So people are trying to use this in early 2020, 2000. In early years, they're trying to establish human embryonic stem cell by somatic cell nuclear transfer. And then this scientist, Han Woo-soo, the Korean scientist who claimed in 2004 and 2005, he published 2 science papers. He said he was successful. He was able to establish human embryonic stem cells by using somatic cell nuclear transfer. If you're interested, check out this scandal, because later on it was proved he didn't. He just mixed some pluripotent stem cell, most likely mouse embryonic stem cell, into his preparation. So he can show pluripotency. He can show chimera and such, what you have already learned. It's a fabricated result, but it's a famous scandal. Unfortunately, in this area of research, in stem cell research, we not only have some of the best scientists who made some of the extraordinary, groundbreaking discoveries, but also we share some of the maybe a higher share, of the infamous scientists. They're trying to gain fame by cheating. Maybe at the very end of this semester, I'm going to give you a few examples. I'm going to talk to you a little bit more about this scientist, his cheating. And now from this point on, I'm going to give you some more technical stuff of how human embryonic stem cells were derived from somatic cell nuclear transfer. The work was done in 2013. That's 7 years after human embryonic stem cell was established in the natural way. So in 2013, finally, people are able to establish human embryonic stem cells by somatic cell nuclear transfer. I want you to keep this question in mind. Why you still want to derive human embryonic stem cell if you can do it in a natural way? Like in 2007, it's already established. You can use in vitro fertilization to establish human embryonic stem cells already. And now, 6 years later, why are you still trying to use somatic cell nuclear transfer to do the same thing? Keep that in mind. Think about that. Later on, we can discuss why this could provide something unique, something that we can use. But for this new advantage, we didn't have it in the natural human embryonic stem cells. But on this slide, I will have to review fertilization and meiosis. I hope you still have this knowledge. Everybody should have learned this in cell biology, or maybe earlier in middle school, in high school. I don't know. But in meiosis, basically, the cells will go through like two cycles of mitosis without cell cleavage, right? To make it simple, I would describe it that way. So basically, at the beginning, the cell will replicate their DNA, therefore to become, but because the chromosomes are not separated, we call it 4C, but 2N. Okay, that's after the primordial oocyte replicate the chromosome. So you have actually 4 copies of the chromosome, but since the chromosomes are still linked with each other, we still call it 2N. That happened in interphase or S phase during DNA synthesis. And then in the first prophase, that's the centrosome are formed, and then centrosome will get aligned to the opposite side of the cell. And then at that time in prophase, you also have the so-called crossover or homologous chromosome recombination, that the chromosomes exchange genetic material. It's called crossover, right? That's what happened in prophase 1. And in metaphase 1, the chromosomes are aligned in the equator of the cell. And then in anaphase, that's when the chromosomes are pulled by the centrosome towards the opposite side of the cell. And eventually you have a telophase. Telophase is when the chromosomes are now located at opposite of the cell, and cytokinesis will start to happen. So the cleavage will make the original primordial oocyte to become one oocyte, and the other is called polar body. This is polar body 1. To distinguish it from the next polar body, we call it polar body 1. So that's the time of ovulation. Ovulation is the process that the oocyte is released to the oviduct, right? It's released from the ovary now to the oviduct, but that is still a rest. That oocyte is still a rest. Usually it is a rest in metaphase 2. So the chromosomes at that time get aligned to the equator, but nothing will happen until that oocyte is fertilized. Until the sperm enter, nothing will happen. When the sperm enters the oocyte, the process will continue. Okay, so you're gonna have the metaphase, arrest, released, you have anaphase, telophase, and eventually that oocyte will become one oocyte, and yet another polar bond, right? Because during meiosis, it's the cleavage is not symmetrical. So you always get an oocyte, the other is called a polar bond. Okay, that's a brief overview of meiosis and fertilization. Why I do that? Because in the discussion that I'm going to give you now, we must have that knowledge. So I'll give you this. I'll give you a slide. I hope you will read this by yourself, paying attention to when the polar body is generated, when the first polar body is generated, when the second polar body is generated, and also appreciate the change of n number. Because at the beginning, it is 4n. And then after the first cleavage, after the completion of the first meiosis, it becomes 2n. And then once the egg is fertilized, and then the oocyte will complete the second meiosis. At that time, the oocyte become 1n. The oocyte will become 1n. However, you have 1n carried from the sperm. Now the oocyte is actually a zygote. It's called a zygote, and the zygote is 2n. So I hope you still have that cell biology knowledge kept. And now let me explain to you a little bit details about how somatic cell nuclear transfer can generate embryonic stem cells. So this again is done in vitro fertilization clinic. It's always done in vitro fertilization. You can never do that in human. You cannot take the embryo out, flush the embryo out, which is not allowed. So some technical details here is, so first of all, you see there's this, let me point to that place and I explain to you. Okay, so this is the polar body here. You have a polar body there. Okay, and then first of all, what you need to do is take that polar body away. That's what happens in this place. You push the pipetter in there. It's a glass pipetter, very sharp. You push through the zona, and then you take the first polar body away, and then from the same spot, you put in an oocyte metaphase spindle. Oh, I'm sorry. So you remove the metaphase spindle. That's this thing. That's the nucleus of the nucleus of the oocyte. This is the acceptor. Okay, because you always need the oocyte to reprogram the somatic cell. Okay, so this is what happened in that. I don't know whether this is called a donor or acceptor, which is very confusing. But this is the oocyte there, the oocyte side there. You take out the polar body. You also take out the karyoplast, which is the metaphase arrested spindle. The chromosome is condensed. At this point, it's called karyoplast. So you take that out, and now instead of pushing a new nucleus from a somatic cell into the oocyte, a lot of times the scientists only put it out, outside of the cell membrane. The outside part here is called the zona, and then the cell membrane is inside the zona. They push this cell, this somatic cell here, that's the somatic cell, and then they give this reagent called a virus. It's actually a virus. It's called a hemagglutinating virus of Japan. It's a strange name. You're going to have the name in the next slide. So this virus will cause cell fusion. So this somatic cell are going to fuse with the oocyte. That's how this somatic cell get in. So instead of just use the nucleus of the somatic cell, in practice, a lot of times you fuse the egg and the somatic cell, which is equivalent to using the stem cell, using the somatic cell nucleus. Why? Because you can appreciate the size of the egg and the somatic cell. The somatic cell really carries very little cytoplasm when you compare to the egg itself. The egg is is big. Okay, and then after that, you give an electric pulse, and they have some inhibitors in here. These are standard methods that people use in vitro clinic to push this zygote to to go down the developmental route, so they will become 2 cells, 4 cell, 8 cell, and such. Eventually to become blastocyst. So that's the procedure they used. And then finally, they were successful. And in that paper, they described why what they have learned. And they described their detailed procedure. But first of all, you see what they shown there. The most important factor in this success is find the ideal metaphase to human oocyte. So you want this oocyte to reprogram a somatic cell nucleus. So the most critical factor here is find the ideal one. But how? Unfortunately, read the note. Unfortunately, we're unable to identify the best oocyte. This is by chance. They did describe it, generally speaking, if the oocyte is from a young woman, usually works better. If it is from an older woman, not as ideal. But they still couldn't figure out exactly what is going on, because some of the younger women still couldn't give an ideal oocyte for reprogramming purpose, while some of the older ones, however, can still give a pretty good oocyte for the reprogramming purpose. So generally speaking, younger one works better, but still they couldn't figure out what is the critical factor, how to select the oocyte so that you can have a better chance of success. Of course, I think by this time, but now the scientists have learned quite a lot already. They know what is the critical factor. So I'll skip the technical details, which I have already described. I just want to give you that point that the most critical factor is the oocyte. So in that somatic cell nuclear transfer, it is not, I would say that somatic cell genetic material is not that important. Although we're trying to do a cloning, we're trying to, to clone the somatic cell. Maybe we're not cloning the somatic cell. We are trying to clone that individual, in which that somatic cell was derived, right? So we're trying to clone that individual. We take the somatic cell of that individual, but the source or the quality of that cell is not that important. As I said, you can take many cell types, liver, heart, fibroblast, doesn't matter that much. What matters is the oocyte, in which you're going to remove the nucleus, but the cytoplasma quality is super important. But what is there, so we still don't know. So, but we do know that in the cytoplasm, you have a lot of things. You even have genetic material, such as the DNA in mitochondria. The mitochondria have its own genetic material. I don't know if you know that or not. The mitochondria will have genes that can encode a few proteins and a few RNAs. So they have their genetic material. So with that, you can appreciate that although the majority of that, maybe the genetic, most of the, I don't know what the correct word. So the genetic material is from the somatic cell in the somatic cell nuclear transfer, but the oocyte does contribute a little of the genetic material. It's from the mother side. I'm trying to give you some some background. So in the next lecture, I will explain will become easier. So the oocyte does contribute a little bit of genetic material from the mitochondria, but the oocyte carries a lot of materials in the cytoplasm. I would say, for the same somatic cell, if you use two different sources of oocyte to do the reprogramming, eventually, if you're generating an animal, let's say the animal is not human, if you're going to generate an animal, I bet those two animals are going to be slightly different, or maybe even hugely different. Although the genetic material are the same, but you use different oocyte to do the reprogramming. So the oocyte does contribute quite a lot to the phenotype eventually you're going to get. So this is the last slide. As you can see, I'm trying to cut the content to go within maybe 40 to 45 minutes. Let's see if I will be successful. So on the last slide, I'm going to describe to you that scientists have already started to appreciate that histone modification is a barrier of reprogramming. Here, the title said embryonic development following SCNT impeded. So that's why you cannot generate a human embryonic stem cell easily from somatic cell nuclear transfer. That is because after that reprogramming, that embryo will have a developmental arrest, will be arrested. It won't be able to give rise to blastocyst. So what's the problem there? The scientists have learned that naturally in fertilized egg, the genetic material, I mean no matter it's from the sperm or from the oocyte itself, you have two nuclei that give you 2N. At one point of development, that's right after you have fertilization, the genetic material will be wide open in terms of epigenetics. I don't know if everybody have learned genetics. In genetics, you learn the concept of epigenetics. So basically, the cells are identical for one species for their genetic material. That's the gene, right? The gene sequence are all the same for a given species. However, in different individuals, in different tissues, the gene expression pattern are different. For example, like my heart will express a lot of genes that's responsible for generating force, muscle proteins. However, in my liver, a lot of genes will be open to encode for enzymes used for digestion, for metabolism, and other things. So different cells perform different functions. So they use some genes and they don't use some genes. For the genes that they don't use, they added epigenetic marks like histone modifications. They modify the histone so the cell know this area I don't care. This area will never get expressed in this type of cell. So in reprogramming, that's a barrier. So in a somatic cell, you have some genes that's expressed and some genes are not expressed. You have different epigenetic marks. But in oocyte, in fertilized eggs, at one developmental stage, all the genes are open. I probably shouldn't say all, but the vast majority of the genes are open. That means they bear the mark that tells you can transcribe from here. Of course, in the fertilized egg, you don't use all the genes, but somehow the cell make all those genes to be open, to make every gene to become possible to get expressed. Although as a consequence, as a matter of fact, maybe not all the genes are expressed, but the possibilities you can express all the genes. But in a somatic cell, it's not a possibility. Only some genes are expressed, some genes are repressed by epigenetic marks. One of the epigenetic marks is called histone lysine 9 trimethylation. So you have 3 methyl group attached to the lysine residue of histone 3. H3. So with that mark, that region are not subjected to expression or activation. So that's different, between fertilized egg and a somatic cell in a nucleated egg. So the scientists now trying to add some histone demethylase. Histone demethylase means the enzyme will demethylate those regions. So to make the methylation mark to get removed. When they apply this kind of a approach there, they add demethylase there, they found it's easier. You can have a higher efficiency in generating blastocysts from SCNT, and of course, you can generate more human embryonic stem cells that way. And the final word of today's lecture is advantage of human embryonic stem cells out of somatic cell nuclear transfer. It is very useful for simulate disease, modeling disease. Why? Because if somebody, for example, have diabetes, it's going to be many genes involved for diabetes, right? It's not a single gene. And if you want to generate a model of it, naturally you can take, you can make two people to mate and eventually give rise to that cell, but most times only one is a patient, the other is not a patient. Even if the other party is a patient, now it's a mixture. Things get even more complicated. But if you use somatic cell nuclear transfer, you're replicating the genetic change or epigenetic change in that patient only. Okay, so that's why this has advantage over just using the natural way to generate human embryonic stem cells. Now you're using the advantage, or if you look from a different angle, this is how SCNT get criticized, because this is cloning, but now you're using cloning to your advantage. Okay, that's the end of today's lecture. And if you have a question, you can stay behind. Otherwise, you're free to go. Thank you.
So essentially, it doesn't necessarily matter where you get the nucleus from when you're doing the somatic cell nuclear transfer, but what really matters is the oocyte more than anything else, and the quality of the oocyte. Is that what I understood?
Yes.
Okay.
Yeah.
All right. And so I did have one more question in terms of how
let me take back what I said. I guess if you do an experiment, I would think like maybe some cells are easier to get reprogrammed. People never know, like when you compare a liver cell, a fibroblast, and a heart cell, which one can have an easier time get reprogrammed, which is possible that there are some differences, right? But if you compare to the quality of the oocyte, it's just a small factor.
I see. Okay.
Yeah, the difference in those somatic cells are smaller, much smaller factor.
Okay. And
yeah, your second.
And so the reason that histones typically would impede somatic cell nuclear transfer, I assume that's because their role in protecting DNA and keeping it condensed, right?
No, I don't know if we'll have learned epigenetics. I hope we will have a chance to describe that. Maybe not far away from now. So the epigenetic marks are modifications on either histone or DNA. It could be histone modification, histone acetylation, methylation, ubiquitination, and DNA methylation. Basically, all those modifications can occur, and a different modification contribute differently to whether that region is open to transcription or closed to transcription.
Yeah. Okay.
It's the case by case. Different modifications suggest different things. Just in this case, the K9, the lysine 9 trimethylation is known to be a mark of repression.
Yeah. Okay.
Yeah, but in the future, if I say K4 methylation, that's lysine 4 methylation, which is actually an indicator of openness.
Okay. Okay. That makes sense.
Okay.
Yeah.
Mhm.
Alrighty. Thank you.
You're welcome.
Speaking of epigenetic, I had a question about gene imprinting. I was wondering, based on my knowledge, whenever during embryogenesis, the gene imprinting methylation is completely erased and rearranged by random chance. So, what does this mean in terms of cloning? Does that mean that the methylation pattern for each of the clone are different?
You know, no matter is imprinting, but imprinting is just one. I would say quite a drastic phenotype in epigenetics. So they basically they make one chromosome not getting the gene from one chromosome, either the paternal or the maternal, not get expressed. They only express the gene from the chromosomes of one side. Sometimes from the the father side chromosome, sometimes it's from the mother side chromosome. It's not about chromosomes, just about single individual genes. Okay, so the process is not random. It's not random. It always on certain genes. Like for example, IGF2 is an imprinted gene, but IGF2 is always expressed from the father side of a chromosome, not from the mother side of a chromosome. Well, another gene that's called H19 is always expressed from the mother side chromosome, but not the that side of the chromosome. So during reprogramming, as you said, I'm glad that you're learning by yourself. So you described that the epigenetic marks are largely erased at the reprogramming stage, but naturally speaking, during fertilization, when you have a zygote, at that point, the majority of the epigenetic marks are also erased because in both oocyte and in sperm, the mature oocyte and sperm, they still have some epigenetic marks. But during that process, those marks are erased and later rearranged, like I said. But the rearrange is not random. It's always about some
Yes.
genes get methylated, some are not methylated. It's not random. But at the very beginning, generally speaking, most of the genes are open.
That makes sense. Thank you for your explanation.
Yeah, when they become more and more differentiated, when they become specified, and then you start to see a lot more epigenetic marks on them.
Thank you.
You're welcome. Hopefully, I will have a chance to describe that. That's fascinating. I will describe that part in our lecture.
I think that would be really interesting.
Yeah.
Dr. Liu, I was
Yes.
wondering then, so when you said like it gets more differentiated, right? How does it know which cell it becomes, for instance, like if it has to be a cardiomyocyte, how does it know that the histone markers have to go to a certain gene and in like inhibit those gene from being translated?
Wow, that's a good question, but I don't think we have an answer. So basically, the cells start to determine their fate quite early, like during in blast, just a little bit later than the stage of blastocyst. What you have is a process called gastrulation. That's the formation of the three germ layers. I would think like a few years ago, people feel like the cells are determined when you start to see the harm start to form, but actually it's not true. Even at very early stage, like gastrulation, you only have three germ layers, but some of the cells has already determined to become cardiomyocyte, or some cells has determined to become blood cells. But
Yeah.
but at that time, maybe they still have some flexibility. If you run short of blood cells somehow, and maybe
Ah,
the cells can differentiate toward that lineage. In early stage, they still keep some flexibility, but amazingly, way before you see a heart, quite a lot of cells has determined to become a cardiomyocyte. And then they start to build the gene expression profile. They start to mark the regions to get expressed, and some regions not to get expressed, and then they continue to build that. So, progenitors,
yeah,
from
yeah.
the germ layer, the mesoderm to become heart is still a long process, although
True.
it takes only a few days in mouse, but in human it take a little bit longer. But that's a that's a many steps involved.
It
In
looks
each
like
step, it's going to involve histone marks,
yeah.
DNA marks to make some genes turn on, to make some genes turn off.
So would you say that there's possibly like some kind of like protein where we don't know whose function is basically allowing for those histones to be reprogramming the system, the cell?
Well, actually, we know, but it's so
Oh.
complicated. We know it's so complicated. There are techniques that we learned at the beginning. It's called chip sequencing.
Yeah, yeah.
You can always study the marks in nowadays, even in individual cells. That's what scientists are. That's a hot area. So basically, the scientists they take the earliest embryo and then they isolate the cells into individual cells. They do sequencing, RNA sequencing, histone modification sequencing, DNA modification sequencing, and then they tell you, they build a map like throughout the genome. Because the genome is always the same, no matter at what stage. It's the same same DNA sequence, and then they describe maybe at this one cell stage which region is open, which region is is closed.
That's crazy.
It's very crazy. They describe this. Hopefully, maybe in a few years, we know that throughout our body, all cells get sequenced.
Yeah, yeah. Hopefully.
Yeah, that will open up a lot more opportunities for us to appreciate disease.
Yeah, yeah, 100%. Thank you.
You're welcome.
Professor, I have a question.
Yes,
So you said that even though two clone individual that come from the same somatic cell could turn into two different individuals, right? Sometimes.
it's not to turn into different individuals. Like what I said is, you for the somatic cell, you take the nucleus.
Mm-hmm.
That somatic cell can be any somatic cell of choice.
Yeah.
Yeah, but I feel there must be difference in the. I mean, the cell source could make a difference during reprogram.
Mm-hmm.
Yeah, because different cells will have different epigenetic marks. If the during reprogramming, that marks are not erased and reprogrammed completely, and you can predict the original cell source will make a little bit of contribution to the traits of the individual born out of the process.
Yeah, because
Because
I think like for the egg, it's not only contribute the mitochondrial DNA, but maybe something like transcription factor or something in there
yes,
that
yes,
also contribute the different.
that's true. Maybe you have a very good point. Maybe the from the the donor, the egg donor,
Mm-hmm.
they could have people from different ethnic groups, and could be like a very tall and powerful one, and there's others not that you know as powerful, things like that.
Yeah.
That does make difference. Like you said, maybe there are transcription factor abundance, or even the they have different transcription factors in the oocyte that can
Yeah.
all contribute to the reprogramming efficiency. I'm glad you're thinking along that line.
Yeah, because I remember about like embryonic carcinoma and then embryonic stem cell. They just different because we induce different transcription factor in there. So
Okay.
I think like the transcription factor play a really important role in here.
Yeah, it's transcription factors are proteins, but in the old days we feel like transcription factor is the most important factor in cell fate, like to determine what cell the stem cell will become, but not anymore. And now we know not only proteins, including transcription factors, but other things like microRNAs, non-coding RNAs, they all play a part in cell fate determination.
Yeah, and I also have read about morphogene, like a chemical. The concentration of some chemical also determine the differentiation of the tissues.
Yeah,
Morphogene.
yeah, I see what I mean. But in oocytes, let's say that maybe not. It's all possible. It's all possible. Maybe it's just a metabolite, for example, right?
Mhm.
And maybe in that one you have more fatty acid that can provide more energy or something like that. They all contribute to the process. So as scientists, we are trying to appreciate it is what is the most important factor in the oocyte.
Mm.
Yeah, we can eventually maybe we can rank. There are many factors there which
Right.
plays absolutely the most important role, which plays a slightly low not as important things like that. Yeah, but as you know, we still this is still early, in terms of understanding that process.
All right. Thank you, Professor.
You're welcome.